I'd like to welcome all of our listeners today. Thank you for joining us. Today we are speaking with Dr. Gail Mandel, who is joining us from Portland, Oregon. She's at OHSU, and she's joined by two of her lab members. Gail, I'm going to let you introduce Saurabh and Dan.
Okay, Saurabh Garg on my left and Dan Leoy on my right. They are postdocs in my lab, and they are co-first authors on the gene therapy paper that we're going to talk about today. They did all the work, and they work together and bounce ideas off each other. And when we do double-blind experiments, they switch so they don't know what they're looking at. So they're a good team.
Yep. Very, very important, the double-blinded. We're hearing about, all these papers coming out about the importance of rigor in preclinical testing. So, okay, so we're talking today about a gene therapy paper that has been funded, the work's been funded by RSRT, that deals with delivering healthy copies of the MECP2 gene in an attempt to slow down the disease progress and reverse some of the symptoms. And the paper's being published in Journal of Neuroscience. Congratulations. So I wanted to just start with a little bit of intro. Gail, I've known you for a long time now. I think we first met in 2000 at a meeting that Alan Wolf organized right after the MECP2 gene had been identified. It was in Washington, D.C., and took you a few years before you started doing Rett Syndrome Research. What finally got you interested?
Well, for many years my lab has been studying other kinds of repressors that are important for neurodevelopment. And we noticed when we started looking at where our repressors were binding to genes, and when we started purifying those complexes, that MECP2 would turn up in some of those biochemistry experiments. So we initially thought that maybe there was some relationship between MECP2 and the repressor we're studying. And so we started to test that hypothesis, and that hypothesis turned out to be wrong. It turns out the reason that we often saw MECP2 with our repressor was because MECP2 is everywhere. Yeah. And so the probability of seeing it on a piece of DNA with our repressor was just chance. Yeah. But by that time I was already kind of hooked in that I thought that Rett was a fascinating disease from a neurobiological point of view, and I thought my lab had good tools for studying MECP2, and we were coming at it from really a very different perspective than, say, Huda and Adrian, who were studying it differently. So we thought maybe we could add something.
Yeah, no, and that's a really important point. I think it takes a lot of different perspectives and ideas and ways of looking at a problem, because this is a complex protein and a complex problem. So let's start talking about the paper, which is important because it really has clinical implications. Obviously, we're interested as a parent and as a executive director of an organization that's trying to find a cure for Rett syndrome. These kind of translational studies are really important to us. So gene therapy's has been around the concept's been around for a number of decades, and the concept is pretty easy. You have a gene that's mutated. Rett syndrome is a single gene disorder, so if we can deliver healthy copies of the gene, you solve the problem. Sounds easy, but there are challenges involved, and gene therapy has had a rocky road with lots of challenges, although now it seems to be in a kind of a renaissance where finally there's a lot of encouraging data on a variety of disorders. So I'm really encouraged. So Gail, you're not a gene therapist. Tell us how you got started in this.
It was Dan's fault.
Thank you, Dan.
Yeah.
Dan was at a neuroscience meeting, and he went to a poster from a graduate student or a postdoc?
From a graduate student of Huda Zogby's lab, actually. And at that poster, there was another student from Brian Kaspar's lab, who is a gene therapist. And he and I, we got to talking, because the three of us were talking, really. and I learned that they happen to have a virus, AAV9, that was able to cross the blood-brain barrier and affect and transfer genes to cells in the brain. And obviously that sent off some bells in my head. And he and I started talking about getting Gail and Brian to talk to each other to try to see what we could do.
Yeah, Dan was very excited. He actually called me from the meeting.
Yeah.
And, of course, my first response, as I always do, is no.
Yeah.
Because I know nothing about it.
Right.
And I didn't know anything about Brian Kaspar, and I'm usually pretty selective in whom I collaborate with. But it had a lot going for it when Dan came back and the three of us discussed it. And so there are kind of two good reasons, in my mind, to consider it for Rett and in our mind. One is that the genetic studies pioneered by Adrian already showed it's a reversible disease, even in late symptomatic stages in mice. So that suggested that you could put back a good copy of MECP2 and possibly reverse or maybe stabilize or just improve by putting it back. And the second reason is that MECP2, whether it's an activator or repressor or changes the chromatin, it binds to the chromatin, and it's going to change a lot of things. Yeah. And so right now I would say we don't really have any single target that we can pick up and say, okay, if we fix that with a small molecule drug, we're going to improve the life of the girls.
Right.
You know, that's not to say that, for example, some of the work John Bissonette and Sarb are doing, trying to find drugs that help respiration. That's not to say that that's not worthwhile, but it's not going to have a holistic
or profound effect on
multiple phenotypes.
Right.
So because it's reversible and because we're dealing with a transcription factor, although it may not be a conventional one, it seemed the most direct way to go for a cure is to put it back.
Right. No, I agree, which is, and I say this to families often, I mean, we can work on downstream targets, BDNF, IGF-1, and chromatin remodeling, HDAC inhibitors, a whole long list of potential drugs. They're not going to cure Rett syndrome. They may improve one or two or a subset of symptoms, which, you know, because the girls have so many symptoms, we would take any improvement in any symptom.
Sure.
But if we really want to see the dramatic cure that everybody talks about, I think for that we have to address the underlying genetic problem. And to do that, you're going to do that either by activating the silent MECP2 or gene therapy or protein replacement, maybe with modifier genes, a combination. So I agree. So in gene therapy, like in real estate, we say location, location, location is really important. So you have to get genes into enough cells and into different parts of the brain. And the way gene therapists do that is with a vector. So can you talk a little bit about, just a few sentences about the various vectors and what some of the pros and cons of them are and what made you decide to go with the AV9? And then we'll get into the results of the paper.
Okay, I'll go for it. So yeah, location does matter. But if you see the symptoms of Rett syndrome, they are mainly neurological or brain originated. But if you see the expression patterns of MECP2, it is present all over the body. Means if you see the expression in lung, it is almost comparable to brain. So it is not that those cells are not compromised in terms of function for the Rett syndrome. So we need a vector, or we wanted to use a vector which put back MECP2 all over the body. So the first criteria is that, and then as most of the symptoms are neurological, you need a vector which can transduce the cells of central nervous system. So we decided using adeno-associated virus 9 because it's a unique property that it can cross the blood-brain barrier. You give an injection in periphery, use the intravenous injection, and then you have the transduction of central nervous system cells. So that is a remarkable property, and we thought utilizing that property is the best way to emulate the function of all the cells in body.
Okay. Do you have anything to add, Dan?
Yeah. Well, I mean, I that's the reason to use AAV9. If you believe that if you take the very basic approach that if MECP2 is there normally, whether it's the brain or elsewhere in the body, and you want to fix the disease, you want to put MECP2 back where it normally is.
Right.
Whether or not we know what it's doing there, if it's normally there, you want to put it back. AAV9 can do that.
And adeno-associated virus also have added advantage in terms of that they don't integrate in the host genome, so they don't cause a lot of side effects like other vectors like lenti. They integrate into the host genomes. They can integrate into the really important locus, which can induce oncogenicity or something like that, or adverse effect, which could be a problem in long term.
Right. So I just want to, lay people may not necessarily understand what that means. I'm just going to clarify that. So some viruses, lentivirus, which also have gotten quite a bit of press in the last couple of weeks because of 2 high-profile papers using a lentivirus approach for 2 genetic diseases. The worry there is that they integrate into the host genome, and depending on where they integrate, they could cause problems. So, for example, they could inactivate a tumor suppressor gene, and then you end up with cancers. And so that's the worry. So that's an advantage of the AAV9. It does not integrate into the genome.
But
Yes,
the other thing is, if I understand those papers correctly, what they're doing is introducing lentivirus into a cell, and then they're injecting the cells
Into the body.
into the body.
Yeah.
So
it's different than what you did. Yeah.
do that right now for Rett
Yeah.
by conventional means, although you and I have been talking about these transplant experiments, which offer the potential for that. But if you take that out, it's different because we're looking for an IV solution, and they're doing a cell, and they can select for what they want in the cells first, and then do the transplant. But that's different.
Those are for peripheral diseases.
Yes, those are the peripheral.
Well, no, they did it for the nervous system too.
Well,
So let's talk about the results of the paper, what you found. Who wants to start?
why don't you do the first part?
Okay. Yeah. So we started with a proof of concept study using the Cre recombinase adeno-associated virus and using the model created by Adrian's bird lab and corrected the MECP2 expression in own locus. So we don't have to worry about the overexpression phenotype in those particular experiments. And we found that reactivation of MECP2 at its own locus had very profound effect on the longevity as well as a lot of symptoms associated with the Rett syndrome in male models, as well as really robust effect in the heterozygous females, where you already have the 50% cells expressing MECP2 and reactivating more cells in there almost corrected a lot of behaviors, including motor skills, then cognition and respiration. So that is the most profound effect on females.
We did this because even though we can't cure a girl with this Cre recombinase.
Yeah, and I'm not sure people. Let me just hold that thought for 1 second, Gail. I just want to make sure people understand. So when Adrian Bird did his experiment, when he wanted to activate MECP2, he injected the mice with tamoxifen, which basically was a way to get the Cre to unlock the gene. Okay.
Yes.
So what you did is you delivered Cre with a vector to basically mimic the experiment to get a benchmark level.
The idea was if we couldn't repeat the genetic experiment with the virus, then
it wasn't worth moving ahead.
The wall. So the idea was if we could show that with the virus we can completely recreate what Adrian did genetically, then that sets the bar for what's possible.
Right.
And then we could evaluate adding MECP2 back.
So I want our listeners to know, stay tuned, because then we're going to talk about results of what happens when you actually deliver MECP2. The Cre experiments help to set the foundation. So go ahead, Gail. Yeah. Or whoever wants to.
So since we were able to see both in males and female mice an improvement with the Cre virus, we said, great, let's see what happens when we use AAV9 to put MECP2 itself back.
Right.
And so we did the same. So we did essentially the same experiment. We injected MECP2.
Same vector.
Same vector. We just put MECP2 in instead of Cre. So this really is a potentially therapeutic mode of delivery.
And we changed the promoter to its own promoter. Yeah.
Yeah,
So-
people won't know what promoter means.
So we used all genes are regulated by the surrounding DNA sequences. The amount of protein you're going to get from a gene is dependent on what is around the gene in the genome. We took a piece of what's known to control, at least in part, the expression of MECP2, and put that into AAV9 along with the MECP2 gene itself. So there was a piece of its endogenous promoter there.
Right. And you can't put everything in because you're limited by the amount that the vector can. This is a Trojan horse, really, and it has a limited capacity of what it can take on board.
Exactly. And so we put what we could in and asked what happened. So we injected it to the mice, and we saw so the Cre virus infected. It caused MECP2 to be re-expressed in roughly 50% of the cells in the brain. However, when we did the MECP2 virus, we didn't reach that level of re-expression in the brain. We were more in the range of 30% of the cells in the brain gaining MECP2.
Mm-hmm. So
in the females, where you already have some cells expressing, you have 50% of your cells expressing roughly.
Right.
If you add 15% more cells, what we saw was an improvement in multiple motor parameters, in growth rates, and we also used a phenotype scoring system that takes into account overall condition, tremor, hind limb clasping, which is a mouse phenotype for Rett, seizures, and in all those parameters, we saw marked improvements. What I mean by that is some of those symptoms stabilized and other symptoms actually reversed, and that was quite incredible. And I think, and I can go on and on about it, but I think there's some questions later that I'll address those things.
And one thing that I think is important is, these were symptomatic females, right? There were females that were 7 to 10 months old, which means
absolutely.
So, and it's important for parents to realize that. Sometimes there's experiments where scientists are able to rescue the symptoms from appearing in the first place. Those are quite different than taking existing symptoms and making them better.
Right. Yeah. And that's obviously for girls that already have Rett.
Yeah, that's what we need.
That's exactly right. Mm-hmm. So we were able to inject symptomatic females and see improvements, which is very, very exciting.
So there were two kind of take-home messages for me. One is that you get very good results just with 15% extra cells, which is kind of surprising, I think. And do you? You're nodding your head. You weren't surprised, Dan.
Well,
it's good news. Let's put it that way. It's good news because now you could try to increase that percentage.
Right, but
that's the idea.
Absolutely,
that's the idea.
Because I think it's important to point out that one of the important phenotypes for girls with Rett is respiration. And mice with mutated MECP2 also have respiratory problems. And even though we were able to correct a whole battery of motor symptoms to wild-type levels, to normal levels, we were not able to get as clear-cut a result on the respiration.
As when you did the Cre.
Right. And that's probably, we had very few mice in that cohort. So it's hard for us to say really what the result there is, but it's not clear-cut that it improved like all the other motor parameters. And that may be a reflection of, for some symptoms, for some phenotypes, you need more cells corrected than 15%.
Yeah.
So, and as far as it being surprising, if you think about it, males already have 0% cells expressing MECP2.
Mm-hmm.
Females have 50% roughly cells expressing MECP2, and compared to males, they're much better off.
Yes.
So there's a dosage effect. The more cells if you can get MECP2 back in 100% of the cells, that would be ideal. But there's not necessarily an a priori reason to think that you need that to improve the phenotype from the condition of the girls if you can just add some cells above 50%. Yeah. And that's really what our results would say.
Could you envision, say that best efforts still yield 15 to 20%? In theory, and I know you're not medical doctors, so but in theory, could you do the gene therapy procedure twice?
Yeah, we've talked about that. We're not gene therapy people, but it's something that we've even thought of trying in the mice.
Right.
Yeah.
So take the ones that have already been done, give them another dose, see what happens. Yep.
I think I have a feeling outsiders would worry about. Then they would really start to worry about the overexpression.
Yeah.
Or not even that, but the
immune
and liver, that kind of thing.
Yeah. Immune response from the body.
So we really have to measure liver enzymes and make sure that the double dose isn't having other side effects. That's probably even more important.
So this brings me to the second take-home message for me. And one of the reasons that some scientists in the community were not big fans of a gene therapy approach for Rett syndrome is because of too much the possibility of delivering too much MECP2. So in some cases, the amount of protein doesn't matter if you have too much, but in Rett syndrome, there's a question about that because we know from the MECP2 duplication syndrome that having twofold, and also from mouse studies, we know that having twofold more MECP2 can lead to a neurological disorder, which is quite serious. So when you deliver gene therapy, you can't, in most cases, you can't regulate how many copies of a gene go into a cell. So there was that worry. But you didn't see signs of toxicity from overexpression, and that's good news also.
Yeah, and we did single cell fluorescent approach for that, and we did not see overexpression in males as well as females. And we were surprised about that, why we did not see the overexpression in females, because they already have some 50% cells having MECP2, and if you are putting on top of that another extra copy, you should see overexpression. But in our single cell fluorescent measurements, we did not see any overexpressing cells or a population of overexpressing cells in females. So that was a bit surprising for us, but I think this is something we don't understand yet, and whether this is something to do with the females versus male biology, or it is the function when we are topically expressing MECP2 through this particular vector. It is something that it has some regulatory mechanism that it is keeping MECP2. Yeah. Mm-hmm.
Or maybe having too much MECP2 in an adult organism is different than having too much from conception. Maybe going through development with an
and that's important. I'll throw this out there too, that there's another study that was done using AAV9 to deliver MECP2, and in that study they did see overexpression when they put it into a wild type, a normal mouse that already had MECP2, but importantly, they did not see any adverse effects of that overexpression, and those were adult mice. So it might be that the overexpression concerns have a very strong developmental component.
Right.
We really don't know.
Okay. So, so what are the next steps? How do we go from your paper to a clinical trial?
Yeah. So I think we think that maybe the first thing to do is try to optimize our vector system so that we can get more than 15%. Mm-hmm. I think that's important to try. And then I think we can probably do that. And then if we can, then we would want to go into preclinical and toxicity testing to look for adverse effects seriously in the mice. Look at liver enzymes. And there's a standard protocol for preclinical tests, and we would do that. So I think for the gene therapy approach, that's what we need to do. We need to focus on the vector first, get a better vector. I think all our experiments and those of others, the other AAV9 group, they're very promising, I think.
Yeah.
But we're not there yet. We all agree we've done a proof of principle. But now we have to make it better if we can.
So Gail, you're part of the MECP2 Consortium that RSRT is funding with Adrian Bird and Mike Greenberg. And can you tell us a little bit about how that experience has been and kind of having people to bounce, even though they're not gene therapists either, but having people to bounce off ideas and brainstorm together, how that experience has been?
Well, I've always believed that two heads are better than one for science, just like everything else. People bring different perspectives. I think Mike and Adrian and I truly bring three different perspectives to the problem, but we're all very open to each other's ideas, and we're all very comfortable sort of criticizing each other in a constructive way, and I think that's really important. And it's to be able to share unpublished results early was really beneficial, as evidenced by the fact that we published 3 papers basically in 2.5 years, which is almost unheard of. And not only that, but at least in our case, the paper flew into the Journal of Neuroscience. I've never had a paper accepted so quickly. Little criticism, really. And I think a lot of that is due to input from Mike and Adrian. In fact, Adrian's a co-author on our paper.
Yeah.
And what that allowed us to do was we were not going to test direct injections into the brain. And we don't do that, and we were focusing on the IV approach. Adrian was doing direct injections, and I think he might never have published that had we not been at these meetings and shown him the IV results. So in the paper, we compare the direct injections to the IV, and the IV worked much better. And so I think a natural question for people is, well, what about, why can't you just inject it in different places in the brain? And Adrian and Elaine's work showed that there are a lot of technical problems associated with that with the virus.
Right.
You mainly infect the cells right around the bolus where you inject. There were some secondary problems. So that never would have happened without the consortium. So, and then of course the work of Adrian and Mike just blended really beautifully and came out in 2 high visibility journals, and really I think altered the way people might be thinking about what MECP2 might be doing. It's I think just a wonderful way to accelerate findings.
Okay, so I hope that we gave our listeners a good overview of the paper and what the next steps are and what it might mean clinically for kids and adults with Rett syndrome. Thank you so much for your work, and we look forward to the next paper.
Okay.
All right.
We'll give it our best.
Thank you.
Thanks, Monica.